There are three mechanisms that bacteria employ to transfer resistance. This is sometimes called horizontal gene transfer. Horizontal because it's transferring from one organism, one bacteria, to a different bacteria. It's not through its progeny, it's not through its offspring. Going through the offspring would be vertical gene transfer. So from mom and dad to child is vertical because there's a direct genetic connection there, whereas horizontal is one bacteria has it and gives it to an entirely different bacteria that was not reproduced from it. Our three methods of horizontal gene transfer are transformation, conjugation, and transduction. We're going to go over these and we'll also do some labs on these so that way we can get some experience and see what actually happens. The first one that we're going to talk about is going to be transformation. Transformation is when you have a donor cell, and I know I already show it here as being exploded, but you have a donor cell that's just a normal cell. And what happens is that donor cell will lyse. And so when it lyses, the DNA becomes sheared. The DNA becomes cut up into small pieces because of the different enzymes that exist in its surrounding environment and all that nice happy stuff. And so when it comes out of the cell, it is not protected by anything. It is completely outside the cell. We call that naked DNA. So all naked DNA means is that it has exited the cell and is now in that space outside of the cell. Transformation uses that naked DNA to then be uptaken, to be transported across into a recipient bacteria. There are two ways of doing this in the lab. In the lab, this can be done by electroporation, which is where you're going to do an electric shock in the cells. And so by doing this, or you can do heat shock. Heat shock is brief high heat, and then you bring it back down and into a nutrient solution. And the reason why these work is because normally you have your plasmid or your naked DNA. Because remember, this can be just normal naked DNA that's not attached to anything. Normally, it can't get through the porins. It's a little too large. And so what we need to do is we need to make the porins larger. So you'll notice that on this part, it's too small. The plasmid can't get through. However, by passing electricity through it, it gives it a jolt and causes it to open, and it causes it to open real fast, causing a suction. Heat will do the same thing. We know that heat increases particle motion, which increases space between particles. We're doing that very quickly, and so it causes this to swell and open up, and as it opens up, it will suck in the bacteria, DNA, It'll suck in the DNA, allowing the DNA to make it to the inside of the bacteria. Also by doing this shock, so this was moving it from from the cell outside of the cell, and now here is where we're showing it entering the new cell. So it would be down here that it would be naked DNA because it has exited its donor cell and has entered into the recipient cell. So by using this method in the lab, we're not waiting for the cell to lyse or natural cell death. We are forcing the pores to open up and allow transport through. The second type of antibiotic resistance is the one that we are going to do in this first lab, and it's called conjugation. With conjugation, it's important to notice our donor cell is referred to as the F+ cell. Our recipient is the F- cell, and that's because in order to do conjugation, you need to form a sex pilus. And so a cell with an F+ gene, so it's not just that it's called that, it has a gene, the cell that has that F plus gene can make a sex pilus. That F plus gene, I don't have it drawn here, so let me go ahead and draw it. That F plus gene is actually a gene that is on the plasmid. So there you go, we can see that. And so it's actually a gene that's on the plasmid. What happens is my purple gene there is going to be my ampicillin resistance, so my gene for antibiotic resistance. We write this as either AMP with a dash and an R, or a lot of times how you'll see it on tubes or in publications is with the abbreviated antibiotic and then a superscript of a lowercase r for resistance. So the donor cell that is positive for that F gene, so the F plus, is going to extend its sex pilus. And by extending the sex pilus, it will begin to attach to the second cell. The recipient cell will then be opened, and so now it serves as a tunnel system. And so the tunnel system, the plasmid, is going to unwind. It unwinds at the 5' end first. So it goes 5' to 3', and then it will circle up. And you can see down here that we have our 5' end. The way that it cuts this plasmid is it uses endonuclease to nick the outer strand of DNA. So our inner strand, because DNA is double-stranded, our inner strand in our plasmid, our circle, is going to remain behind in the donor cell, and the one that was nicked, the outside strand, is going to go to the recipient. Once that gets transferred, I'm not sure why my cell is gone, but there we go. Once that is transferred, it transfers both genes. It transfers not only the antibiotic resistance, but it also goes with it, because it was part of the plasmid, is the F gene. And so the F gene means that now our recipient, while it used to be F negative and not able to make a sex pilus, now it can make a sex pilus. And so you transferred the antibiotic resistance and the ability to transfer that antibiotic resistance to other bacteria. That's what was transferred from the donor to the recipient. And you'll notice I say here that plasmids also contain the F plus gene once the transfer is complete. HFR plasmid contains the F plus gene, and the HFR stands for high frequency reproduction. They're reproducing as fast as possible because they want to reproduce with this antibiotic resistance. In conjugation, an important note is that only plasmid DNA is transferred. Only plasmid genes are transferred. The genomic DNA is not transferred. So this one, only the plasmid DNA is transferred. Up here, we could take the naked DNA, or we could take the plasmid, or both. Down here, it's only plasmid. The way that we can use this then is that we can take bacteria. We've got two different bacterial strains here. LB agar is just plain agar, the stuff that you poured in class. And so with that, I put two different strains on. So I have a pink strain and I have a blue strain. And with my pink strain, I can put it on ampicillin plates. And so I'll notice that, well, my culture 1 will grow on the AMP, meaning that it's ampicillin resistant, but my strain 2 does not. And so that tells me that strain 2 does not have ampicillin resistance. So then on the strain 2 has streptomycin resistance. And I know that because when I grow it on a plate that contains streptomycin, the bacteria grows, but we'll notice that strain 1 did not grow. Following the same idea then, that means that neither of them will grow when I have both antibiotics in the same plate. However, what we can do is through conjugation, we are going to put those two different strains kind of overlapping with each other, kind of mixing. And then the bacteria that form in that mixed space have the potential to be resistant to both antibiotics. And so if this is what occurs and we plate this bacteria, then we will see that, and it would no longer be strain 1 and strain 2 because it would be the offspring strand. And so we would see that this new bacteria will grow on our strep and ampicillin plate. The way to determine then, well, which strain transferred the gene, is you'd have to look at where the gene is located. For example, if we say that on strain 1, it is the ampicillin gene that is on the plasmid, and if we say that the streptomycin gene is on the genomic. And so with that, the plasmid has to be transferred to the genomic. There's nothing else that can happen. And so remember, we can't transfer genomic DNA in conjugation. Only the plasmid transfers. And so that means that strain 1's plasmid was transferred to strain 2 in order to create our LB ampicillin our LB ampicillin streptomycin resistant dual antibiotic resistance. The last type is going to be transduction. Transformation and transduction, I always get confused, not gonna lie. We've got transduction. And transduction is we're going to use viruses, specifically bacteriophage. And a bacteriophage is a virus that only infects bacteria. The viral structure is there's a capsid that has a protein shell that protects genetic material. The genetic material can be DNA or RNA. There's a collar that connects it to the sheath, which is a hollow tube that transfers the genetic material. There's a base plate that contains spikes to allow it to attach to the bacteria. And then there's tail fibers. They look kind of like legs, so it's kind of how the virus can move around on the bacterial surface. And so there are two cycles. There are two things that the virus can do. It can do the lytic or it can do the lysogenic cycle. And so at step 1, I show it up here. At step 1, what's going to happen is the phage is going to bind to the bacterial cell. And then when it binds, it's going to inject, so it's going to put that plate down on to the bacteria, and it will inject its genetic material into the bacteria. Once the viral DNA or viral genetic material is inside the bacteria, the prophage will detach. And then the phage DNA will integrate into the host DNA. So the host will uptake it into its own genomic DNA, which means that the host will now make copies of it. And so the bacteria reproduces like normal, but also replicating that phage DNA. And so this continues and makes tons and tons and tons and tons. So this does not result in bacterial cell death. It's just going to propagate the viral genetic material. Eventually what happens is it goes from the lysogenic cycle, and you'll notice that at this location, I have both circles coming in. And so after this happens enough times, it's made enough copies of the virus, then it will enter into the lytic cycle. It is also possible for a phage to do the lytic cycle straight away. So there are two directions that it can come into the lytic cycle, either by exiting the lysogenic cycle after several rounds, or from a initial infection with the bacteriophage. What the lytic cycle does is it takes that DNA, so it might take that bacterial DNA where it's been replicated numerous times, and it will start to package and actually make the virus. It makes the viral capsid, it makes all of the parts, and so now there are new phage and DNA replicated within the bacterial cell. By this happening, the phage becomes so numerous that eventually the cell lyses, eventually the cell bursts. And so with the cell comes the phage as well as the original bacterial genomic DNA. And then the process will start again as those phage go and infect other cells. And so how does this transmit antibiotic resistance? Well, because on the phage itself is that antibiotic resistance. So there's a little part of that phage DNA that conveys that resistance to the antibiotic. And so now all of these viruses are going to go out and they're going to infect other bacteria. And when they do, they're going to transmit that antibiotic resistance. And now all the cells that this DNA gets passed to are resistant to that antibiotic. And this is the transduction method of antibiotic resistance transfer. So there are three types. We have transformation, we have conjugation, and we have transduction.